This program is a presentation of UCTV for educational and non-commercial use only.
It's my true pleasure to introduce our last speaker. Definitely last but not least, the core of our course, stem cells. And last week you had your introduction into human embryonic stem cells, and tonight you'll learn about different stem cells. And this would be the person to ask about pretty much any kind of stem cell, since he's the head of the Stem Cell Institute here at UCSF. So with that, I'm going to really introduce our speaker for tonight. Dr. Kriegstein received his MD and PhD from New York University, then did his residency at Boston Children's Hospital in Boston, then was a professor at Stanford in the Department of Neurology, then moved back to the East Coast to Yale to do both clinical and research work in neurology again, then moved to Columbia, where he had the chance to be involved in the Stem Cell Center, and finally in 2004 joined us at UCSF and became the head of the Regeneration Medicine Institute and the Institute for Stem Cell Research here. I would say you cannot get more cutting edge than what you will hear tonight. And with that, I really don't want to take up any more of your time, and please join me in welcoming Dr. Kriegstein.
Well, thank you for that kind introduction. It really is a pleasure for me to be here this evening and to welcome you all to what turns out to be the final session of these series. And my style may be a little different from some of the previous speakers you've heard. I'm really more interested in hearing, in a way, what you would like to hear. And I'm happy to talk about the science, about the promise of stem cell research, about the politics, the controversies, any of the aspects that interest you. I have slides, of course, prepared, and so I will be happy to start and go through most of my slides. But it doesn't matter to me if we finish them all or not. I'm more interested in having a bit of a dialogue, and in the end, I'd be very satisfied if you all walk away with a better understanding of what the interest and the excitement about stem cell research is all about. So please feel free to raise your hands or interrupt as we go along with questions, and consider these slides really just a framework for our discussion. And again, it's a very personal view of stem cell research, so I'm happy to give you my biases and tell you where they are. I thought I'd start just by reviewing a little bit about what stem cell lines are all about. And the most controversial and perhaps the most useful cell lines are the ones that come from humans, either from human embryos or, as we'll talk about in a few minutes, other sources of human cells. And the label of embryo is sometimes misleading. And so I wanted to show these images, all of which are embryos, as we go from left to right. These are human embryos. The one on the left is a single-cell embryo. This is just the fertilized egg, essentially. And on the right, you can already see an embryo that has taken shape. I mean, there are limbs, there's a head, there are organs that are developing already. Those are all embryos. Beyond the right-hand image, which is 6 weeks old, is the fetal stage of development, where, again, the organs are even more developed. So when we talk about an embryonic stem cell that comes from a blastula stage embryo, we're really talking about the cell the image that's right in the middle there, the 5 to 7 day old embryo. So in those 5 days or so, it's gone from a single cell to a cellular ball. All those cells that you see in that image in the center, these cells have all arisen from this original fertilized egg by a series of divisions and divisions and divisions that have gone on for several days. And at that point, there are no limbs, there are no organs, there's clearly no heart, no brain, no liver, nothing like that. It's just this ball of cells. And it was thought for a very long time that these cells were all equivalent to each other, that they all had the potential to be any cell in the body. And that's a term that we call pluripotentiality or pluripotent. And to show you a little bit more about what that looks like, here's a ball, a cluster of these cells, which are sitting in the head of rather the tip, the sharp end of a pin. So these are obviously highly magnified. And what that embryo looks like at that 5-day stage is probably smaller than the period at the end of a sentence. So this is a microscopic ball of cells. And it's from those cells that one derives a stem cell line. And so to show this schematically, here's the embryo at 3 to 5 days, cut in half. So the cells on the outside are now shown as this ring of cells. And in the middle, there's a cluster. They're shown here darker. This is known as the inner cell mass. And these cells, in the normal course of events, would eventually become the entire body. The cells on the outside that form the circle would actually adhere to the uterus, and this would form the placenta. So these are, sort of extra embryonic. These are the structures that are not going to be part of the embryo. So those have already been segregated. Or they've become two different distinct cell types. And it's this inner cell mass that ultimately gives rise to a stem cell line. And that happens only, of course, if you put it in the right tissue culture environment with the right kind of growth factors and stimulate division of these cells. And what will happen is these cells will divide and divide and divide and divide and eventually form a colony of cells, all of them identical to each other. And this is what defines a stem cell line, the fact that the cells will divide forever, that is, in the right conditions indefinitely, and they'll self-renew, so that at every division, the daughter cells are identical to the cell you started with. Question. Right, so the question is, would these be classified as adult stem cells? And that has to do with the controversies having to do with the Bush doctrine in 2001 saying that you couldn't use federal funds to study embryonic stem cells created after 2001. We'll get to that in a minute. No, the answer is that these are embryonic stem cells because they come, as I've just described, from a 3 to 5 day old embryo. And in order to create this cell line, you really have to destroy the embryo, and that's where the controversy begins. So you can't, at least initially, years ago, you couldn't just remove a single cell and create a colony like this and leave the rest intact. In order to make this colony, you really had to take those cells and dissociate them in a dish so that the embryo was essentially destroyed at that stage. And so these stem cell lines were created from embryos that were frozen at this stage, which is part of the process of in vitro fertilization. These are clinics that are all over the world now. There have been millions of babies born through in vitro fertilization, but there have been tens of millions more embryos created that aren't being used. And so many of those are slated for destruction. That is, they're frozen away. Families decide they've had enough children, they've got excess embryos left behind, and what they decide is to destroy them. Or, in cases like these, they can donate them for specific purposes. So here at UCSF, we have consent forms that allow families, if they want, to donate embryos that would otherwise be destroyed for stem cell research. And when that happens, we can take these embryos and turn them into these embryonic stem cell lines. That's been done at UCSF. It's been done in many other centers around the world. And so those are called embryonic stem cell lines because they come from human embryos. I hope that's clear because this is really the heart of the ethical controversy. Now, once these cells are renewing forever in a dish, they're not terribly useful that way, but what you can do with these cells, under the right conditions, is you can turn them into any cell type in the adult body. So you can take these embryonic, undifferentiated, so-called pluripotential cells, and in the right conditions, make them into brain, skin, muscle, lung, heart, or even germline cells like a sperm or an egg. And that hasn't quite been done yet, but we can talk about that. So these cells then have the potential to become any cell in the human body. And in that way, they become the building blocks for this whole new approach to regeneration medicine, ways of rebuilding organs or replacing cells that are injured or dead or dying in diseases. And so that's the therapeutic promise of stem cell research. And this is pretty much what fueled the whole interest in stem cells 5 years, 10 years ago. But things have changed very quickly in the last few years, in fact. And there are new kinds of stem cells that I want to make sure you all understand. First, there are the adult stem cells, which that gentleman referred to. These are found in not the embryos, but in the adult organs in the body. So, for example, in the placenta, you may have heard about cord blood stem cells. In the bone marrow, there are blood-forming stem cells. And in the brain, even, there are stem cells. So these are cells in the adult body that are dormant most of the time, except in places like the bone marrow where they're continuously active. But under the right circumstances, they can generate the cell types found in those adult organs. So the brain stem cells can make brain cells, but they can't make heart cells, they can't make liver cells. The stem cells in the liver can make liver, but they can't make heart, they can't make brain. So they have limited potential. They're not pluripotent like the embryonic stem cells are here. They have a limited potential, but on the other hand, if you're trying to make replacement neurons for a disease like Parkinson's disease, you don't want to make heart cells. You don't want to make bone cells. You really want to make just this specific kind of nerve cell. And so for those reasons, it may be useful, actually, to have adult stem cells. But they're not as plastic. They're not as pluripotential as these embryonic stem cells. And the other limitation up to now is that they don't self-renew indefinitely. These adult stem cells seem to have a shorter lifetime. Even in the right conditions in a laboratory, they'll divide multiple times, but eventually peter out. So they may not give you, in fact, the large number of cells you might need for cell replacement therapies. Question. This is an interesting question. It has to do with the fact that anything other than a 5-day-old blastula is considered, in quotes, an adult. So you can actually derive stem cells from a fetus, and people have, and companies have, and they're being used in clinical trials, which we can talk about in a little while, probably will talk about in a little while. Even though they come from fetal tissue, which I showed you on that continuum is just to the right of the embryo, they're still considered adult stem cells. Now, you might wonder, how can a fetus be considered an adult? And it's a bit of an artifact of the distinction people have been making, the important distinction they make, between this stage embryo and everything else. So something that comes from an adult organ, and placenta is pretty much considered an adult organ, is considered an adult stem cell. And aside from which, the cells that come from the placenta, the stem cells from the placenta, it's unclear whether they actually arise in part from the mother or, in fact, entirely from the fetus. But nonetheless, there are stem cells in the placenta that generally make placental tissue, but in the right conditions can make blood-forming cells of all types, and so they can be used the same way these bone marrow cells can be used therapeutically. Now, more recently, there's another way to make stem cells, and that's to convert adult cells, and these are not stem cells, these are adult, what are called somatic cells. These are differentiated cells, like, for example, a skin fibroblast. You can take those cells in the right conditions in a laboratory, convert them into stem cells that are just as pluripotential, it seems so far, as these embryonic stem cells. But there are some major significant differences between this way of making stem cells and this way of making stem cells. First of all, there's no ethical issue. We're not destroying embryos to take a sample from the skin and convert it into a stem cell. There's no embryo involved at all. The way you do this, by the way, is to introduce genes into the cell. In fact, so far it looks like 3 or 4 specific genes that just by themselves, in ways that we don't fully understand, will reprogram this adult cell back into this undifferentiated, pluripotent stem cell-like condition. A very magical thing that we still don't fully understand, but it works. And it can convert not just skin cells from the surface skin, but fibroblasts from other organs. You can take a cell from the liver, as we mentioned earlier, not a stem cell, but a fibroblast from the liver, or a fibroblast from the pancreas or some other part of the body, and use those cells to make stem cells. And just in the last few months, it's been shown, in fact, it was just published last week, that these cells are not entirely the same as the embryonic stem cells because they have a memory of the organ in which they arose, where they came from. So if you take, for instance, a fibroblast from the liver and make it into a stem cell, it'll reproduce indefinitely. But when you try to make it into these different cell types, you'll find it's much easier to turn it into a liver cell than a nerve cell or a bone cell. And similarly, if you take this from the nervous system, from the brain or the spinal cord, and make it into a stem cell, although you can force it in these other directions, it's much easier to make it into a nerve cell type than any other kind of cell. So these skin cells or these other adult cells that are converted into stem cells are being explored in great detail now to see how they compare, how different they are, how much better or worse they could be for certain kinds of purposes compared to these embryonic stem cells. So currently, the science tells us that we need to study both embryonic stem cells and these so-called induced pluripotent stem cells to see which ones are going to turn out to be most useful for certain applications. So right now, places like our institute are comparing these two cell types with a great deal of interest. Now, being able to take an adult cell and convert it into this induced stem cell type not only gets around this ethical issue, but it makes two important things possible that couldn't be done with these embryonic stem cells. First, you can take these cells from a patient, somebody who has, for instance, a genetic disease, say Lou Gehrig's disease, ALS. You can take a skin biopsy from that patient, in a laboratory convert it into these induced stem cells, take those stem cells and make them into the nerve cells that are dying in Lou Gehrig's disease, these motor neurons in the spinal cord. And we actually know how to do this, and this has been done with human skin cells, turn them back into motor neurons, and then study them in the dish in the laboratory to figure out what the difference is between normal motor neurons and the motor neurons from a patient with ALS. So you're therefore studying the disease, the human disease, with human cells in a laboratory. And this is a much more powerful approach than studying some sort of animal model, like a mouse model of the same disease, because you're actually working with the human cells that are suffering from that disease. And if you can find some way of treating that disease in a dish, the chances are that it'll work much better in a patient than it would if you were just treating, say, a mouse cell in the laboratory. So these become very powerful models for certain kinds of diseases. Question? Right, so the question is, can they be used for any human being or just from the patient they were taken from? Well, that's another aspect to the advantages, actually, of having this way of making the stem cells. And that is that if you were going to treat that same patient with those cells, for example, if it's a patient who has Parkinson's disease, we'll talk about later, where a certain kind of nerve cell is missing, it's dying, you want to replace those dying cells. And you'd like to be able to do that in a way that doesn't trigger immune reaction or rejection. As you may know, organ transplants, are life-saving, but part of the problem is that they bring with them the cost in the sense that the patients need forever to be on these immunosuppressive drugs that have their own problems and a whole range of very serious side effects. Well, if you can take the skin sample from the patient himself, convert that into the stem cells we're talking about, and then make them into the neurons that you're going to then put back into the same patient, because it's the same patient and the DNA is exactly the same, it shouldn't create the same kind of immune rejection as it would if it came from somebody else who's not related to that patient. So this gives you a patient-specific treatment that might avoid these immune rejection problems. So that's the advantage of taking a skin cell from a patient and using it to treat the same patient. On the other hand, you could take a patient's skin cell, turn it into a stem cell line, and create huge numbers of nerve cells and use them to treat other patients. And that would be equivalent, essentially, to taking an embryonic cell, which comes from someone who's not related to any of those other patients, making those into stem cell lines and using them to treat patients. So in that sense, even though it may come from one individual, it's really no different than a stem cell that comes from one individual embryo. It could be used to treat lots of other patients. Now, I should mention these therapies, in terms of these induced pluripotent stem cells, are theoretical right now. And the reason for that is that in order to convert them into stem cells using these 4 factors, the way those factors are introduced most of the time is by what are called retroviruses. And these retroviruses introduce genes into the genome, into the DNA of these cells, but they do it by inserting into random spots in the chromosome. And when they land and insert into those spots, they actually create a small mutation. Now, many of these mutations might be silent. They might not actually create any mischief. But it turns out that more often than not, if you just randomly insert these mutations into the genome, one or another of them could increase the chance of uncontrolled cell division, namely cancer or tumor formation. Not only that, but it turns out one or actually 2 of these factors that are used to make this magical conversion are themselves oncogenes, which are genes that, in fact, produce cancer. So for those 2 reasons, because of the insertional mutagenesis, as it's called, and because some of these factors can, by themselves, increase the chances of cancer, we don't think that any of these approaches will be used to treat patients with the cells. Nonetheless, right now, that's the most efficient way to convert skin cells into stem cells. And so these procedures are being used in laboratories all around the world right now to make disease models, because if you make a model of a disease, you don't really worry so much if it creates cancer. If 1 in a million cells become tumor forming, you can get rid of those in a laboratory. That's not a problem. But you wouldn't want to use these to treat a patient. Having said that, there are some rapid advances in ways of getting around this insertional mutagenesis step. So there are soluble drugs that can take the place of some of these genes. At least 1 or 2 of them have been discovered and published so far. There are other ways of introducing these genes transiently, so they're not integrated into the genome, so they don't cause mutations. All these methods seem to work, and there's now been 4 or 5 workarounds that seem, in that sense, clinically safe. The problem is that they're all very, very inefficient. So the number of cells you start with and the number of cells you wind up with is really grossly imbalanced. So it doesn't work all the time, and it takes a lot of effort. The most efficient way still right now is to do it with these 4 factors. But that could change next week or next month. I mean, the field is moving extremely fast, and there's enormous excitement about this whole approach. In fact, as many of you know, it's only been a year since this was first done with human cells. And now it's being done, all over the world. There are publications, as I mentioned, in ALS, where patients with the disease have been used to generate human stem cell lines. Those have been turned into motor neurons. And papers have already shown that they're different than motor neurons from patients who don't have the disease. So these things have been happening, just this last few months, in fact. So the field is moving extremely fast. And it wouldn't surprise me in a year from now, there's a very efficient way of making these cells that doesn't introduce any kind of genetic alteration. And that would make it clinically safe. But right now, that's not the case. So we'll have to tune in again, maybe next year. So you probably heard, I think, last week from Susan Fisher, who's here at UCSF, and she's actually been able to make stem cell lines not from that 3 to 5 day old embryo, but even the 2 or 3 day old embryo, much smaller, that's called a blastocyst, at a stage when there could be 8 or 16 cells. And she's been able to take just, as you can see here, 1 cell, and in the right conditions, could create a colony of cells, and then eventually a cell line from an individual blastocyst. This is a true clonal cell line because the single cell has now made the entire population of maybe millions of cells. Not only that, but at this early stage where there may be only 8 cells, again, it was thought originally that these were identical to each other, that at that stage, by the way, you can have twinning in normal course of events. That is, if in normal development, one of these embryos at this stage is cut in half or cleaved, the 2 halves of that embryo have the ability to reproduce themselves and regenerate 2 complete individuals, and you wind up with twins, identical twins. So at this stage, it really seems as though these cells are identical to each other. But what Susan has found is they're not, and that if you take these 8 cells and create a cell line from each one of them, each one of those cells will have a different, slightly different ability to make certain types of tissues than the other cells from the same stage embryo. And so even at this early stage, there's already some genetic distinctions between these cell types. Now, they can be erased, apparently. You can make pluripotent cells out of any one of these. But there are some echoes, some memories, I guess, of what cell type this was supposed to be that still persist even through the creation of a cell line and then de-differentiation down a different pathway. So these blastocyst embryos at this early stage, or morula embryos, may be very much like those skin stem cells we just talked about. They all seem to have a little bit of a different memory. And that may explain why it seems to be more and more important that we have large numbers of these cell lines, because some of them turn out to be easier to use for certain purposes than others. So if you're trying to make a treatment for diabetes and you want to make insulin-producing cells, those come from tissue known as endoderm. And certain cell lines from maybe the right blastomere seem to be better able to make endoderm than others. Or if you want to treat Parkinson's disease and you want to make neurons, there's certain cell lines that are very good at making neurons, but not so good at making bone or muscle or pancreas tissue. So, there's a lot of diversity here that we don't fully understand. That's true from the embryonic cells, as also true from the skin cells that I talked about, or the adult cells that come from other tissues of origin. So these are schematics that basically show what I've just talked about, how you can make an embryonic stem cell line from this early morula or blastocyst embryo and create unlimited numbers of these pluripotent cell lines. And these are, each one of them called a cell line. We can now, So the field has moved so quickly that I completely forgot about something known as cloning, or therapeutic cloning, or sometimes referred to as SCNT, which stands for somatic cell nuclear transfer. This was all the rage about 4 or 5 years ago. And many of you may remember this fellow from South Korea who got into trouble because he claimed, well, it was fraudulent. He made these fake figures and published a paper in Nature claiming that he was able to make a clonal cell line in human patients using this method, SCNT. And it was really exciting at the time because it had the potential to make patient-specific cell lines. In fact, at the time, which was only 4 or 5 years ago, it was the only way people knew to make patient-specific cell lines. And the trick there was that you took a cell from the adult, it could be a skin cell, you remove the nucleus, and the nucleus, of course, contains all the DNA, all the inheritable material, all the chromosomes, and you injected the nucleus into an egg that has its own nucleus removed. So you have to start with a donor egg, in this case a human egg. And you'd have to remove the nucleus of the egg first, and then insert the nucleus from this adult skin cell. Then you'd have to activate it in a certain way to get it to start behaving like it was a fertilized egg and divide and divide and divide and become this blastula embryo, which you then treat just like the embryonic cells we talked about earlier to make a cell line. So from here on, making the cell line is the same as right now pretty much old hat. The trick is this part, taking the nucleus, getting an egg, combining them, and getting them to start behaving like an embryo. Now, this bit of magic is done routinely in the veterinary world. The first example, of course, was Dolly, the cloned sheep, and since then, lots of different species have been cloned. Humans, on the other hand, have never been cloned. That is, nobody has ever been able yet to take these nuclei from a skin cell, put them into an egg like this, and create an actually viable embryo. So this process, known as somatic cell nuclear transfer, has never been done in humans. But it was claimed to have been done by this fellow in South Korea, and the reason it was so exciting is because it allowed you, if it was true, to make a cell line from an individual patient. Now, I've just told you we can do that now with skin cells, and we don't have to use embryos. We don't go through this method at all. We short-circuit the whole thing by going directly from the skin cell to the stem cell line without making embryos, without using eggs, without any of this stuff. And so the science has moved by so quickly that what was one of these goals that everybody was trying to achieve 4 or 5 years ago is now passé. Nobody that I know is trying to do this anymore because there's no point. It would be more difficult. Getting donated eggs has been a huge hurdle. It's very difficult to get healthy eggs donated for this purpose because most people who harvest eggs like this want to use them for IVF to make babies, not for this kind of research. Besides which, most places have decided that paying women for these eggs for research purposes is not ethical, and therefore if they can be paid for IVF work and not paid at all, they have to donate them for research, the choice is usually very simple. So very few eggs have been donated for research purposes, even while programs like this at Harvard and UCSF were in effect 4 or 5 years ago. So just an example of how quickly things have changed. Shinya Yamanaka, by the way, is the person who was responsible for this change. He created this method of inducing pluripotent stem cells from skin cells using those 4 factors. And he won the Lasker Award in 2009. Everyone assumes he'll win the Nobel Prize pretty shortly. He has a laboratory in Kyoto where he did most of his early work, but he was a postdoc at UCSF at the Gladstone Institute, and those were the happiest years of his life. And he was very motivated to come back. In fact, he has a laboratory that he established 2 years ago at Gladstone, and he spends 1 week out of 4, that is 1 week every month, here in UCSF Gladstone. And the rest of the 3 weeks he spends back in his lab in Kyoto. So you see him here on the campus from time to time, but he's a very famous fellow right now. And we have established, also just in the last few months, a facility that actually routinely makes these iPS cells. And this is just an example of how the science, again, has not only moved so quickly, but it's been disseminated so rapidly. So that we and now lots of centers around the world routinely make these cell lines. So we have physicians who have patients with rare diseases they want to study in a laboratory. They've been pretty much using mouse models up until now. But now if they can get a skin biopsy from their patients, they can give them to this facility, which we have here on this campus, and within 4 to 5 weeks, they can actually get several cell lines, human embryonic stem cells, converted into these stem cell lines with their patient's DNA, with that particular disease, which they can then use in the laboratories to study the mechanisms of that disease. And they can also be used, by the way, for drug screening, for high-throughput screening procedures, to develop conventional drug therapy for these diseases. The therapies may not be stem cell therapies, but they'll be made possible by the use of stem cell biology to create these human disease models that you can study in a laboratory. And this is happening already. We can give you several examples maybe if we have time as we go through these slides. So what stem cell biology can do today, or at least what the promise is right now, is that we can take these stem cells or induced pluripotent stem cells from individual patients. We can use them to model diseases, as I've mentioned already. We can use them as ways of delivering payloads to certain parts of the body. Some of these stem cells have the ability to what is known as hone in to tumors, for example, and deliver a payload to a tumor. Or in the nervous system, they can migrate and integrate into circuits, and they can deliver enzymes that are missing in certain kinds of metabolic diseases. So we can give you some examples of that way that these stem cells can actually deliver genes or therapy or growth factors to parts of the body. They can obviously be used to study fertility and human development. For cancer treatment, there are several things about cancer and stem cells that you should know. One, as we described already, is that some stem cells, because they can indefinitely self-renew, have the ability to form tumors in the human body. So in most cases, if you're trying to use a cell-based therapy to treat a disease, you probably want to not use the stem cells, but you want to first turn those stem cells into a well-behaved, non-dividing cell, like a nerve cell or a pancreatic cell or a liver cell or some other kind of cell type that's not a stem cell. So the trick there is that if you start with stem cells and make them into nerve cells, you may have a few cells in that mix that are not fully differentiated, that haven't become the nerve cells yet, that may still have, some of the abilities to divide. And even if you have a few of those and you give them to a patient, if he's a young enough patient and he lives long enough, they may form a tumor. So as a safety concern, there's a lot of interest now in somehow eliminating all the cells you're going to deliver except for the ones that are well behaved. You want to get rid of all the stem cells. Any self-renewing cell type you don't want to inject into a patient if you can avoid it. And that's actually the concern that's limited any trial so far that I'm aware of in the country or in the world using embryonic stem cell derived cells. So you may have heard of a trial that Geron Corporation had gotten FDA approval to do in spinal cord injury. So this was a trial that was planned to enroll about 6 to 10 patients within a couple of weeks of spinal cord injury and to treat them with embryonic stem cells made into oligodendrocytes. And we'll talk about oligodendrocytes in a little while. But they're the kinds of cells that insulate nerves. And so they're not nerve cells, but they support the nerve cells. And there's a way of actually making large numbers of oligodendrocytes from embryonic stem cells. And that method was pioneered by a scientist who licensed that technology to Geron. And so they have a way now of making large numbers of oligodendrocytes, a specific cell type that they think will help patients with spinal cord injury. And they were planning to put them into these patients. And the FDA had given them approval. But in the time it took between the approval and the enrolling of the first patient in that trial, they had studied more mice, and they discovered in a few of these mice what they call cysts, but I think are probably better termed, small tumors. Now, these aren't the kinds of metastatic tumors that you might think of that are spreading all over the body, but they are unregulated cell growth in the cells that they injected into these mice. And so the FDA, so they informed, as they should, as they're obliged to, inform the FDA of these mice that had the cysts, and the FDA then put the trial on hold. It's still on hold. And, Geron is now doing more experiments in mice, trying to understand why this one batch of cells behave the way they did. And if they can explain that and they can satisfy the FDA that they understand what's going on and it won't happen again, then the trial may go off hold and start again. But that's, in the U.S. at least, the closest trial so far to using embryonic stem cell derived cell types as a therapy. But the risk is just what I've described, the fact that they could form tumors. And so that's a real concern. Now, there's another disease trial that's getting organized in the UK using embryonic stem cells to make a kind of cell type found in the eye, known as a pigment retinal epithelial cell. Now, these cells are targeted in a disease known as macular degeneration or age-related macular degeneration, and they're hoping to use these cells in a clinical trial this year or early next year. And the indications are that the UK will give them permission to do that. If that happens, it'll be the first trial, because the Geron trial is on hold, the first trial using embryonic stem cell derived cells in patients. Now, the safety feature there, I know this is going to sound a little grim to some of you, is that they put these cells in the eye. And they're right in the back of the eye where you can see them under the right kind of ophthalmoscope or the right kind of eye exam. So they can monitor them. And if they start forming a tumor, you'll be able to see that. And the hope is you can remove it, either remove the cells or, worst case scenario, remove the eye, that is, enucleate the eye. That obviously will save the patient, but at the cost of their eye. Nonetheless, that's considered, I think, the safety valve for that trial. And you may hear about that over the next 6 months to 8 months to a year because, as I say, approval for that trial is pending. So that's the relationship to cancer. I've already hinted about drug discovery. That is, you can use these cells, especially the iPS cells, to model a disease and then study the mechanisms of the disease or test drugs that might actually rescue or protect the cells that are dying in diseases. And these are, of course, studies done on the human cells so that the drugs you discover are more likely to actually succeed when they get to clinical trials. The other thing that's happening already is the use of these stem cells for toxicity screening. Every drug that's marketed in the United States, or in that matter probably most of the world, has got to pass toxicity screening for liver toxicity and heart toxicity. And these cardiotoxic effects and liver effects are usually screened for in animals. You know, you have lots and lots of mice or rats, and they give them all kinds of injections of these drugs at high doses and look for effects. Well, those animal studies, mostly in rodents, you know, don't always mimic what happens when you put them into patients, into human beings. But now you can take stem cells, create unlimited numbers of, say, liver-type cells or cardiomyocytes, heart muscle cells, and you can expose those cells in a laboratory to, you know, whatever doses of these drugs you want to use and look at the effects on those human cells. So you can now test toxicity of drugs using human cells rather than, you know, animal models to see whether the drugs are going to work or not, or at least if they're going to have toxic side effects. And then, of course, for regeneration medicine, the idea that these cells could be the building blocks for regenerating whole tissues or organ systems. And that's still a ways off, and that involves a partnership between the biologists and bioengineers and materials science people and so on. But that's happening as well. And so there are already some artificial organs like bladder, for example, that have been used. You may have heard about trachea replacements that are a combination of mechanical parts and stem cells. So regeneration medicine is really just at its infancy right now. But the hope is that these stem cells will really make that possible. So now these are cardiomyocytes. It turns out not to be very difficult to take embryonic stem cells or stem cells that are induced pluripotent stem cells and turn them into cardiomyocytes, which, you know, are the contractile muscle cells in the heart. And if somebody has a heart attack, the cause of the heart attack is usually the vascular issues. You know, the blood supply to the heart muscle is compromised. But the effect is that the heart muscle dies, and heart muscle doesn't regrow. And so the real problem, congestive heart failure and so on, that occurs following heart attack is because the heart muscle is irreversibly injured. And the hope is that one can actually turn these stem cells into cardiomyocytes like this, and then get those cardiomyocytes to graft into the parts of the heart that are still healthy and replace the injured part of the myocardium. So that's, you know, again, the dream or the hope of treating patients with heart attack. And we're getting closer and closer. So, you know, we can make these cardiomyocytes, and you can see that they're all synchronous. They're coupled up to each other, and they're synchronously beating. The problem so far has been that no one yet has been able to get them to integrate into the rest of the heart so that they beat together with the heart. So, you know, it doesn't help if they're beating asynchronously, if they're not hooked up with the rest of the heart muscle. So the issue now, one of the big challenges, is to get these cells to actually couple up with the existing cells. And there are strategies to do that. We have a partnership with some physicians and scientists in Israel. We're using proteins, overexpressing these proteins that actually couple these cells to the existing cells. And that seems to work in dogs so far. Hasn't been done yet in human cells. But we and others are working toward the hope or to the eventual goal of getting muscles like this, heart muscles made in the dish, to act as patches to treat heart that's been injured following heart attack. You can also, by the way, make blood vessels that way, and so the hope is that you can maybe also replace the blood vessels that are inflamed and that are causing the problem in the first place. The other thing that's happening already is that these cardiomyocytes that I've just showed you can be made from patients who have cardiomyopathies. These are inherited metabolic disorders of heart muscle. You can then study those disorders in the heart muscles in the dish. And this just shows that you can actually look. Here's an EKG of a patient who has a prolonged QT interval, a certain kind of arrhythmia, and that same arrhythmia is reproduced in cells in the dish that have been grown from his skin cells. So you take a skin cell, make them into an IPS-induced pluripotent cell line, take those and make them into cardiomyocytes, and those cardiomyocytes, in unlimited numbers now in the dish, display exactly the same cardiomyopathy that the patient has, that his heart cells have. So these you can study, you can poke them, you can do whatever you want to them because they're not in the patient's own heart, they're in your dish in a laboratory, but they exactly mimic the condition that the patient has. So this is an example of how you can model, in this case, a cardiomyopathy and start looking at drugs that might actually improve the cardiac rhythm and discover, therefore, therapies that will help the patient. And these are conventional drug therapies, but they're being discovered because of the use of these cardiomyocytes in the laboratory, made possible through, you know, the breakthroughs in stem cell biology. Now, at this point, I was going to take a little bit of a detour and talk about my own area of interest, which has to do with brain development. But I could take a break for a second, and I know there haven't been a few questions as I've talked the last few minutes, so I'm willing to ask any of you who have questions to ask them now rather than at the end of the hour. So anything so far about stem cells that are confusing or that aren't fully understood or that you'd like to explain, me to explain a little more? Right, a good question. So it has to do with how much of what I've told you is already happening and how much of it is still just potentially happening. And I hope I gave you the impression that it's happening right now, or we're really at that moment where it's just starting to be translated, because that's exactly where we are. So the toxicity studies, there are companies that have been that have sprung up, that are created now around the intellectual property-based, that deals with making liver cells that you can use for liver toxicity screening, cardiomyocytes that can be used for cardiotoxicity screening. And so these companies are already hard at work getting this to happen. So there's venture capital invested in this. There are scientists and laboratories working very hard to achieve those goals right now. No one has yet made, as far as I'm aware, and again, these things change very quickly, but as far as I know in the published literature, no one has really made a bona fide hepatocyte, that is a liver cell, that you can use to do enzyme studies and so on for liver toxicity. People have come close. You can make cells that will produce certain kinds of enzymes that the liver produces, but none of them yet are perfect matches to hepatocytes. So we're getting closer, and I wouldn't be surprised if in a year or two someone doesn't achieve that goal. But to give you an idea, that's where we are right now. Don't quite have hepatocytes. We do have cardiomyocytes, as I've indicated, and there are ways of making them in large numbers and making them in pretty pure, in high degrees of purity. And once again, there are companies that are actually starting to market these cardiomyocytes for cardiotoxicity studies. Laboratories in individual academic centers like ours, and the cells I showed you, by the way, came from Bruce Conklin's lab here at Gladstone, are already using IPS technology to make cardiomyocytes from patients with inherited diseases and using those as vehicles to study the disease, and for high-throughput screening to see if they can come up with drugs that can correct those, in this case, cardiomyopathies, the arrhythmias. So those kinds of high-throughput screens based on cells like this, that's already happening. I don't want you to go away thinking that you can use these cells for any disease, because if you take a skin cell and turn it into a cardiomyocyte, it'll only have a disease if it's an inherited disorder, if it's a disease that will be carried in the genome, in the DNA of that patient. If somebody acquires a disease based on, say, smoking, let's say you smoked a lot and you've got a cardiomyopathy as a result of that, you can take a skin cell from that patient and make it into a heart cell. It'll be a healthy heart cell because it won't have had the effects of 40 years of smoking or whatever. And so that won't be a way of modeling that disease. So you have to choose the right disease. You've got to choose a disease that has a genetic component, or else it won't be mimicked, or it won't be transferred to the cell line. Yeah, that's a good question. How about autoimmune diseases? Well, of course, that's a whole immune system, that's formed by cells, actually stem cells that continue to produce immune cells throughout your life. And as you probably know, in certain kinds of cancers, you can actually replace, to some extent, the immune system by ablating the immune cells and replacing them with stem cells that then reconstitute the immune system. So, so there's been a long history of experimentation, as it were, with humans and the immune system and the stem cells that make those immune cells, those circulating immune cells. The thing about immunity is that, as I mentioned earlier, it's a double-edged sword, sort of like cancer in a way. It's both an opportunity for using stem cells to treat patients. On the other hand, when you graft cells into a patient that doesn't come from that patient himself, there's the risk of rejection. And the way to get around that is, again, to modulate the immune system. And there are some interesting opportunities that exist. For instance, you can take advantage of the in utero environment. And we have one of our members of our stem cell group here, Tippy McKenzie, who's an expert at fetal surgery and also an immunologist. And you can tolerize the yet-to-be-born patient to cells that you introduce into the fetal environment. So if you can diagnose, and you can in certain conditions, certain metabolic disorders in those fetuses, you can actually then take cells that you could use to treat that patient once they're born, introduce them into the fetal environment, so that when the baby's born, it will actually be able to tolerate a graft of those cells, or from that particular donor cell, after birth. And you can then try to correct the beta thalassemia or the sickle cell disease or any of a number of these diseases after birth, having tolerized that particular patient in utero. So that's just the strategy that's actually being explored right now for how to treat certain kinds of conditions and take advantage of what we know about the immune system. A lot of the diseases, that we'd like to be able to treat with cell replacement are actually based on autoimmunity, like type 1 diabetes. It's one that you hear about a lot when people talk about stem cells. And the reason is that the cells that are dying in that disease are the islet cells that produce insulin. And, if you could just replace those cells, you could, not have to use extrinsic insulin injections and so on. And that's not just hype. That's actually happening. So a group of UCSF investigators in partnership with a company called NovoCell in San Diego have in the last few years shown how you can actually take embryonic stem cells and under the right laboratory conditions turn them into islet cells. They are cells that can secrete insulin and do that in a regulated way. That is, they secrete the insulin in response to what the blood sugar levels are in the patient, or in this case, the animal model. And these are made from human embryonic stem cells. So they're now hoping to get a clinical trial going within the next few years, and they have a large grant from the state through the California Institute of Regenerative Medicine to do just that. Now, they're making these cells from embryonic stem cells, so they have two issues. First, I'll get to the immune system in a minute. First is the issue of tumor formation. And the strategy they want to use to get around that is to put these cells in a matrix. They actually have a kind of capsule that they can embed them in, and the capsule keeps the cells contained, so the cells can't escape. However, blood and fluids can circulate through. So you can take this implant and put it into the abdominal cavity or into other areas where there's circulation of blood and fluids. And these cells will regulate the amount of insulin based on the amount of sugar that they see. And they can actually take, at least in animal studies, they can make these animals euglycemic. That is, they can regulate their insulin release perfectly. And if they develop tumors, the idea is that you could detect that and remove the implant and possibly replace it with a new one. So, that would be the safety valve in case there's a tumor that forms. The other aspect has to do with rejection. The reason these patients had diabetes in the first place is they had antibodies that attacked their own islet cells and destroyed them. So the issue is, if you give them new islet cells, why won't the same process happen to destroy those new islet cells? Well, it may not destroy, that is, their antibodies may not attack the extrinsic, the islet cells you give them from another patient, because they may or may not recognize them, and reject them the same way they rejected their own. That's one issue. Second issue, even if they did, you could always, in this case, replace them with a new one if you had to. And the other thing is that there is a cell-based immunity that involves this immune cells, circulating immune cells, actually contacting the cells that they destroy. And if the islet cells are in this matrix and planted in the body, that matrix keeps the cells from escaping. It also keeps the immune cells from getting in. So they may actually be protected from some of that rejection because they're in that capsule. So these are things we'll have to learn about when we move to our clinical trials. That's why they're called clinical trials. But it's a strategy that may get around that autoimmune thing. Another disease that, as a neurologist, I'm interested in is multiple sclerosis, which is, again, an autoimmune-based disease where these oligodendrocytes, the cells I mentioned earlier in spinal cord injury, those are the cells that are dying in multiple sclerosis because of this immune response. So we could replace the oligodendrocytes, but the problem is, how do you prevent them from being attacked the same way the original oligos were by these antibodies? And, that's a problem. So diseases like those will probably require, like diabetes and like MS, will require a two-pronged approach, replacing the lost cells with replacement cells, and then some way of modulating the immune response to protect not only the existing cells, but the ones you're giving them, the new cells. So it's a kind of repair mechanism plus some way of arresting the disease, which will involve modulating the immune system. So it'll probably be, a combination of both effects. But, the patients with end-stage type 1 diabetes, no matter what you do to the immune system, unless you replace their islet cells, they're never going to really, have the cure. They'll always require extrinsic insulin, and that'll never be regulated the way as tightly as the actual cells will regulate it themselves. And so I think most people in the field feel that stem cell replacement therapy is part of the long-range approach to type 1 diabetes. The question is, the stem cell lines that are being created here at UCSF, can we get federal funding to work on those lines, and how does that work now? And of course, in the Bush years, things were different than they are now. In the Bush years, in 2001 in August, Bush decided that all those lines that had already been created could be studied with NIH funding. But he wouldn't allow NIH funding for any newer lines that were created because that would require destroying embryos, as I'd mentioned. And he didn't want to be in the business of endorsing or supporting any activities related to destruction of embryos. And so that meant that any of the newer lines, which were always being improved, that is, the technology was improving significantly, none of those lines, which were newer and better in ways we could discuss, could be studied with NIH funding. And NIH, funds most of the research that goes on in this country. And so that was a major, major obstacle to progress in this field. And it led to this response in California where, citizens actually voted for Prop 71 and created the California Institute of Regenerative Medicine in order to do what the federal government wasn't doing, to support research using embryonic stem cells. And so I should mention that the NIH didn't make it illegal to do this work. It just made it, impossible to use federal funds. So it could go on in laboratories that are private, for instance, in industry, which continued, Generon, for instance, other companies were doing this, and academic institutions that had either foundation support, like the Juvenile Diabetes Society or the Michael J. Fox Foundation, or the state, for example, the CIRM, or other states like in Wisconsin and in Connecticut and New York. They had their own state-funded agencies that supported the creation of new cell lines. Now that Obama's taken over, the Democrats have changed these regulations. They've created a new registry which is based on ethical grounds, not some arbitrary line in the sand, but lines that are derived with the correct informed consent, where the patients knew what they were doing and so on, using embryos that would otherwise be destroyed. Those kinds of cell lines would then appear on the registry and could then be used by anyone. And that's the way things are now. There are over 70 such lines on the registry. It continues to grow. Some of the original lines that were allowed by the Bush administration turn out not to have been created with the correct form of informed consent. And so it's been a big struggle that some of these lines that people have been using with federal funds for 8 years did not appear on the new registry because they didn't make muster. No one could find the informed consent, the forms themselves. In some cases, the forms turn out to be woefully inadequate. They didn't even give permission to use these cells to make cell lines. So ethics didn't have anything to do with it apparently in 2001. And so now there's been a big struggle to try to grandfather in these lines because people have learned a lot from using them, and it would be a real waste if you had to do it all over again with new lines. And so just in the last few weeks, two of the most often used lines created by YCell, a company in Wisconsin, are now on the approved list. But they had to really work around these ethical guidelines in order to make them look at least like they were sanitized. So it was a real struggle. Going forward, all the lines that appear on that list, will have passed muster in terms of ethics. Now, I should still say that we're in a paradox where the NIH will fund experiments that you do with the cells, but still won't fund the creation of the cell line itself. So once a line has been created with other funds, you can then apply for registry approval, and then you can use NIH funds to study it. So can anyone in the world. But you can't use NIH grants to create new cell lines. So Susan Fisher, for example, who's running our human embryonic stem cell program here, continues to make cell lines, but she can't use federal funds for that work. So she, in her lab, still segregates the activities that are federally funded from those that are not. And with 2 laboratories and 2 sets of equipment and so on, just to make sure that there's never going to be an audit or whatever that causes trouble, she really segregates those activities. So that's the kind of situation everyone was in 2001. Most of us now who don't make our own cell lines, are happy enough to work with NIH-approved lines. But in those cases where you need to make new lines, you still have to get outside sources of funding for that. That's a long-winded answer, but that's the situation today. Yeah, that's an interesting question. The question is there any advantage to taking tumor cells, for instance, and using them to make cell lines and studying them? Well, so again, one of our investigators here, Robert Blaylock, who's in the urology department, came here. He actually wasn't a urologist. He was a pathologist at Harvard. But he was interested in prostate cancer. And he did exactly what you suggest. He took prostate cancer cells and used them to make a stem cell line. And then he took those stem cells and made them back into prostate cells. And the question there was, would they have cancer? That is, would they be cancerous prostate cells once he made them back into prostate cells? And the answer is no. They behaved like perfectly normal, healthy prostate cells. In fact, the animal he used it to make mice, and the mice, after several generations were perfectly healthy mice. They didn't have cancer. And this is one of the pieces of evidence suggesting that kind of cancer actually didn't form by mutations in the genome. Because if there had been, this is the theory that's prevailed for quite a long time, that cancer often arises as a result of accumulated mutations. So, over your lifetime, some cells will divide, and when they do so, they run the risk of mutations forming, and then over time, you accumulate these mutations, sort of like those retroviruses I mentioned that randomly cause mutations. Eventually, that will lead to some sort of an overburden that produces a tumor-forming cell. And then you take those tumor-forming cells and study in a laboratory, they will have the mutations that will produce cancer. They will have the oncogenes turned on, or they'll have something like that. That was a conventional idea. But more recently, we've learned, some of you may know about epigenetics, which are the ways that you can actually regulate gene expression, not by mutations in the genome, but by actually regulating how genes are turned on and turned off. And these epigenetic switches are actually switched or turned on or off also during life, and you can accumulate certain kinds of epigenetic changes. The difference is that if those epigenetic changes cause cancer or something, they're not embedded in the genome. They don't cause, those aren't mutations. And if you then create stem cell lines like we just described with those cells, you'll wipe out those epigenetic changes, and you'll wind up with just the DNA that you started with, which will be, healthy all over again. That's what Robert was able to show in his pancreatic cells, that the pancreatic cancer was not due to genetic mutations, but due to the acquiring of these epigenetic changes, all of which could be wiped off when you just revert these cells into this into this undifferentiated cell type. So that gave us some insights into cancer formation. And it also, by the way, gives you some ideas of how you might treat cancer, because you can actually make these, turn these epigenetic changes on and off with certain kinds of molecular switches that a lot of people are now trying to do in order to treat cancer. I will spend just a few minutes describing the nervous system, that is how the nervous system arises, how the brain forms. And that'll be an introduction to nervous system stem cells, which we'll talk about maybe for the next 15 minutes or so. So what I want to show you is that the nervous system, we already talked about this in a way, consists of two kinds of cells. There are the nerve cells and then the support cells. And the support cells modestly outnumber the nerve cells, in fact. And they consist of what are known as oligodendrocytes or astrocytes. These are glial cells. They help hold the brain together. At least that was the traditional thinking. And during early development, one of these glial cells has this long fiber here. It's called a radial glial cell. And this is a neuron, a newborn migrating neuron that's migrating along this radial fiber. And it was thought once they get to the right place, these radioglial cells disappear. Turns out that that's not entirely true. What my lab and others have shown is that these glial cells actually are the neural stem cells that make the neurons in the nervous system. And this is just some time-lapse images that show this in a mouse, where this green cell is expressing this green fluorescent protein. It's one of these radioglial cells, has this long fiber, and it's going through a division right now. It's dividing right here along this dotted line, which is right in the center of the brain, area called the cerebral ventricle. And when the cell divides, there's a cleavage furrow that forms. It breaks the cell, divides the cell into two cells. And the parent cell is this radioglial cell with the red triangle, and the daughter is the one with the white triangle. And after the first division, both of the cells sort of move away. The parent cell, the one with the red triangle, has this radial fiber, which, by the way, used to be thought, these cells round up before they divide, but they don't. This radioglial cell still has the fiber, even though it's divided. And the daughter cell is putting out little processes. I don't know if you can see one here. And it's going to migrate along the parental fiber. If you notice this daughter cell, we've just lowered the magnification, is going to migrate along. So this daughter cell is migrating along the parent fiber. And so this suggests that there's a clone of cells, that the parent cell and the daughter cells in the nervous system actually form this kind of a familial group. Now this radioglial cell is going through another division. Now it used to be thought that these radioglial cells, when they divide, made more glial cells, and the neurons came from some other place altogether. Nobody really understood. But this really shows that these radioglial cells make both the glia and the neurons. So they are the neural stem cells in the brain. And the daughter cell is not a neuron, it's also a different kind of cell that's dividing. So it's a progenitor cell. Turns out that this cell is not a stem cell. It has a more limited potential. This can only make neurons, whereas this can make neurons, oligodendrocytes, astrocytes. This is a true neural stem cell. It can make multiple cell types, whereas this is a more restricted type of cell. And so this pattern of neurogenesis, where the neural stem cell makes a daughter cell that has a more restricted potential that makes neurons, that's typical for any organ in the body, that you have two kinds of cells, the stem cells that make everything and the daughter cells that are very limited in what they can do. So this is the pattern that was discovered by us and others about 5 years ago to be the way that nerve cells are created in the mouse brain. And since then, we now have markers that we can use, like this red one, which is selective only for these secondary type of more limited progenitors. So now we can actually tell them apart by these different antibodies in the laboratory. It's a pretty straightforward scheme, but it's not the way things happen in the human brain. And it's only recently that we've started looking at how this compares to human development, that there have been some striking observations or differences. So now we've got markers, only in the last few years, that allow us to distinguish the two kinds of progenitors, the stem cells, which are in red, and the more limited progenitors in green. And this is how it looks in the mouse, and the developing mouse and rat. Now, in the human, there's a similar, around the same zone in the deep part of the brain, there's exactly the same pattern. The neural stem cells in red, and the intermediate progenitors, the secondary daughters, are in green. And it looks exactly like the rodent. I mean, this is, from an evolutionary perspective, this is how it all began, looking pretty much the same. But what we have that the mouse and rat don't have is the second zone, which is known as the outer subventricular zone. And this is a huge zone. I'll show you how big it is in just a minute. But it contains these red and green cells, which don't exist in the mouse or the rat. Now, obviously, it's not surprising that the human brain is different than the mouse and the rat brain. But I show you this because up until now, everybody has been modeling diseases of the human brain using the mouse brain. And when you do that, you miss, all of these cells. So we've started looking at these cells to see what are they and how do they compare, to what's not in the mouse. First, I want to show you how many of these cells there are. So this is the inner subventricular and ventricular zone. This is what the mouse has, the rat has. What we have that the mouse doesn't have is this outer subventricular zone here, which is a huge proliferative area. When I say proliferative area, it's filled with these stem cells and progenitor cells that are making tons and tons of neurons, again, that don't exist, it doesn't exist in the models that we're now using in the mouse and the rat. And so what's in that area, that outer subventricular zone, turns out to be these two very interesting cell types. One in particular is the one shown here. I won't go through all these different stains that I've used, but this is what it looks like. It has a single radial fiber. These only have this one directional fiber. But other than that, they behave very much like neural stem cells. And I'm going to show you a little movie here of these cells. And this is in developing human cortex. And you can see, I don't know if you can follow these cells, they're moving and they're dividing. They jump up and they divide. They jump up and they divide. And they're a highly proliferative zone. That is, there's a lot of cell division going on there. And these are 4 of these newly discovered cell types. They're called ORG cells, or outer subventricular zone radioglia. And you'll see that each one of these cells is dividing in the 20 hours or so that we're filming this, because each one of these cells cycles in about 20 to 22 hours. And they go through cell divisions, one after another, and their daughter cells go through their own cell divisions. And this continues for quite a while, each of the cells dividing and producing daughters that divide and daughters that divide again. And then eventually they turn into neurons. And when they turn into neurons, I'll just skip this because it basically shows the same thing here in schematic. When they turn into neurons, they turn into large numbers of neurons. So here on the left is the rodent radioglial cell that makes a daughter cell that makes a pair of neurons. So after 2 divisions, you wind up with 2 cells in the mouse or the rat. Turns out in the human, you've got the same thing going on down here, but you've got this outer subventricular zone where you have these new cell types, newly discovered cell types that are called ORG cells. And they go through multiple rounds of division, each time producing daughters that themselves go through multiple rounds of division. So you get this multiplicative effect of huge increases in the numbers of neurons that you have. And this, of course, is no surprise. The human brain has more numbers of neurons than any other brain known in the animal kingdom. It's not the biggest in terms of size. It's not the biggest in terms of volume, but it has the largest number of nerve cells. That's the key feature that we have that nobody else has, not even the other monkeys that we're related to. And then in the end of neurogenesis, these cells disappear. So these neural stem cells disappear except for 2 parts of the adult brain where they continue. One of them is called the subventricular zone that my neighbor here at UCSF, Arturo Alvarez-Bulla, discovered. And the other is an area of the hippocampus known as the dentate gyrus. And in those 2 areas of the brain, there are adult neural stem cells that continue to make neurons throughout life. Problem is that, those are very small areas. The subventricular zone neurons are going to the olfactory bulb, so they don't help you in any other way except with smell. And the hippocampal cells are involved possibly in learning and memory. That's what it looks like right now. So that is very helpful. And then as you get older, unfortunately, the number of these cells declines, and that may be related to your age-related memory loss that people my age always seem to have. I certainly do. And that's probably because this neurogenesis is slowing down. So there's a lot of interest in trying to stimulate endogenous neurogenesis to try to overcome those limitations. And that's an area that investigators at UCSF and elsewhere are very interested in pursuing. But these embryonic cells that I've been telling you about, for the most part, disappear when all the neurons have been formed in the adult brain. And this just shows during development how big this outer subventricular zone becomes during the period when most of the neurons in the cortex, the neocortex, are forming. So this just shows schematically these cell types. And for those of you who know about the columnar organization of the cortex, the discovery of these new cells actually explains how we can have this big, huge, convoluted cortex. When I say convoluted, it's, the human cortex is folded, where the mouse cortex is smooth. It's folded because you have so many neurons that you have to pack them in more densely, and it's done by folding the surface of the brain. Nonetheless, you also have an increased number of columns of cells. And these fibers I showed you, the neurons migrating along those fibers, these outer subventricular zone cells have more fibers and create more ontogenetic columns of cells and help explain why the cortex, the cerebral cortex, winds up being bigger and also having many more columns than in, say, mice or rats. So I'll skip all of these points because in this audience it's probably not so important. But what that tells us is that there are stem cells in the human brain and that we're going to find in our human cell cultures that we don't know about from the rodent studies we've done so far. It sort of underscores how important it is to be able to study human nerve cells in a dish. Now, you can't do that by biopsying patients. I mean, you can't take nerve cells from donors and take them out of the brain. But because of stem cell techniques, we can now have unlimited numbers of neurons in a dish that we can create from skin cells. So we don't have to take a biopsy of your brain. We can actually take a piece of your skin and turn it into nerve cells in a laboratory and use those to study the disease you may have in your brain. So, that's one of the things that's already happening. And I just want to mention a little bit about Parkinson's disease, which many of you may know. Is shown schematically here why it's an important target for stem cell replacement. Because a small cluster of cells that all are the same, they all make dopamine as what's called a neurotransmitter, and they live in this part of the brain called the midbrain. These cells are dying for mysterious reasons in the disease. And when they die, they normally project to this area called the caudate, and that projection disappears, the cells disappear. There's no more dopamine in this part of the brain. So the strategy most people are using is to replace those missing cells from here by injecting cells in the target area here that make dopamine. So these cells will produce the dopamine that's missing and hopefully cure or correct the disease. So that's a strategy that lots of people are using around the world, and there are some labs at UCSF trying to do that. But my colleagues, Arturo Alvarez-Billia, John Rubinstein, and Scott Baraban and I, have a different approach to this disease. We reason that because when these cells die, the activity in this area of the brain changes. In fact, it becomes overactive. This is an inhibitory area, so when it becomes overactive, it actually slows behavior. So we took this area that's overactive and decided maybe we could correct that overactivity by introducing not dopamine-producing cells, but inhibitory interneurons that will connect to these overactive cells and literally slow down their activity. And so this approach involves getting these inhibitory interneurons and injecting them into animals, in this case, that are made Parkinsonian first. And so how do you make interneurons? How do you make inhibitory interneurons? Well, it turns out that during normal brain development, they're made in one part of the brain, shown here. It's called the medial ganglionic eminence, but doesn't really matter. And in this one little area, which is a factory for making these interneurons, they then migrate all over the brain. So the cerebral cortex is filled with these interneurons, but they all come from this one area early in development. So it's a little factory, shown sort of schematically here, for making these inhibitory neurons that then migrate all over the brain. So in our earlier experiments, that started a number of years ago, actually 5 years ago, we actually took these cells from the embryos at this age, where there's just millions of them here, took them, dissociated them, and injected them into the adult animals that we wanted to treat to see if the strategy would work. So here's the strategy. In this case, for Parkinson's disease, we collected these cells from embryos, and we used cells that were green so we could see them. We then took rats that were lesioned earlier so that they had Parkinson's disease, essentially, that is rodent version of Parkinson's disease. And we then injected the cells into the part of the target area called the striatum that we thought was overactive in this disease to see over time if these inhibitory cells would actually correct the overactivity. Now, first I want to show you that these cells, which are the green ones, distributed very nicely within this target region, which is the striatum, shown here. And if you look at them at higher power, shown in these images, you can see that they've matured into cells that have processes, the way neurons do, that contact lots of the neighboring cells. We recorded from them to show that electrically they were in fact behaving like the interneurons should, and that they received and also made synaptic connections with the neighboring cells. And this is very important because they really became what's called integrated into the circuits. So they couldn't modify circuit activity unless they connected with the native cells, and that's what these cells did. And we then looked at behaviors. Now, we did a whole variety of behavioral tests. Let me just show you the ones I like because they resemble the patients I see with Parkinson's disease. So one of them is a runway test. So this is a rat that's been rendered Parkinsonian. Actually, no, this is a control rat. Sorry about that. We've dipped his feet in ink, and I don't know if you can see the little paw prints left behind on this paper. This is a very low-tech study of what's called a gait disorder. But you've got these paw prints, and you can easily measure the gait by looking at the inter-paw interval. And in these animals that are rendered Parkinsonian, just like in patients, you know, they move more slowly, the gaits are changed, and the distance between the paws is much smaller. After we've grafted these MGE cells and then waited 5 weeks or more, the gait actually improves, and now they've normalized the distance between their paw prints. In fact, the control group, which were not lesion, they weren't Parkinson animals, they were just normal animals, that we've put these cells into the same area, actually had longer stride lengths than the normals. So they actually ran faster down the runways than the normal animals did, which is, I think, actually a sign that we were modifying motor circuits in a way that really affected, you know, the timing of these complicated movements. The other feature I wanted to show you is also typical of patients with Parkinson's disease. If you look at these open fields and measure the activity of the animals for a period of half an hour, normal animals will, you know, wander all around, which is shown by these sort of tracks. But in the Parkinsonian animals, for the same amount of time, they just sort of sit in one place. They don't move very much, just like patients who have the disease. But once we've grafted these cells into that striatum, you know, they once again are exploring all around the fields. So, you know, and this is another rotary stimulation test that's typical for animals that have Parkinson's disease, and once again, it demonstrates that the treatments actually improved the motor behavior. And this mouse work was just recently published. But how are we going to turn that into anything that might be therapeutic? And the answer is, of course, we can't use these cells that come from the embryos. We have to make these cells from embryonic stem cells. And we've been doing that based on work that John Rubinstein and his colleagues have done over the last few years, which has been identifying the genes that are responsible for making the little factory in the developing brain in the first place. So this area where the cells normally derive is an area that John has been studying for a number of years, and he's found, you know, the sort of the code that's used by the developing brain to make these kinds of inhibitory interneurons. So we've taken that code and reproduced it in our laboratory dish by taking human embryonic stem cell cultures and treating them with the same kinds of transcription factors and growth factors in a dish that would normally expose these cells to in the developing brain. And we've wound up making, in fact, the cells that we want, these inhibitory interneurons that are called these GABA cells, which are shown here in pretty large numbers. And these are now human GABAergic cells created in a dish. And we're now using them in these animal models the same way we use the animal cells. And if these work, that is, if they work as well as the animal cells do, then that gets us a lot closer to actually some cell type that we could use to treat human diseases. Now, we wouldn't start by treating patients with Parkinson's disease, but these same approaches can be used for patients with epilepsy, intractable seizures, for instance, because you can graft these cells into parts of the brain that are overactive with seizures or epilepsy, and we've shown so far in animal models that they can do the same thing. They'll inhibit the activity in those areas and actually correct or improve the seizures. So these cells may be useful for those diseases. They may also be useful for spinal cord injury, neuropathic pain, for a whole series of disorders where overactivity of a very focal area of the nervous system is the problem, and these cells can, you know, actually exert this inhibitory effect if you graft them into almost any part of the nervous system. So that's one thing that we're looking at. These are the cells at higher power. These are all these inhibitory neurons that we've made human cells in a dish. Now, our neural stem cell pipeline at UCSF has quite a number of other strategies that are getting toward the clinic that I wanted to let you know about. And this is a project led by Mitch Berger, who's the chief of neurosurgery here. And it's targeting a terrible disease, glioblastoma multiforme, which is a a brain tumor. You probably all know Ted Kennedy died of this not long ago. And it is really an untreatable brain tumor, the most aggressive form. It turns out that neural stem cells, not nerve cells, but the actual neural stem cells, like those radiogliae I showed you earlier, those cells, when injected into the rodent brain, have this ability to home in or actually migrate to areas of inflammation, which in the case of tumor mean the area around the tumor and in the tumor itself. So these cells have the native ability to migrate to those tumors. And so Mitch and his colleagues have decided if they can use these cells to deliver a payload to actually destroy those tumor cells, that this may be a way of dealing with the tumor. So as a neurosurgeon, you know, Mitch can actually debulk these tumors. He does this all the time when patients have large tumor mass, but they always recur because there are microscopic metastases that he can't surgically reach. And the hope is that these stem cells will reach those areas of micro tumor formation and destroy those tumors. So this is a strategy that's not just theoretical. It's actually being driven toward the clinic with the help of a $20 million grant from the CIRM. And so the goal is that within 3 to 4 years to have an IND, that is a filing for the FDA, to approve this strategy to treat actual patients in a clinical trial. So you may hear about that over the next few years. Another disorder, a neurological disorder, that's actually where a clinical trial is ongoing right now at UCSF, is in this disorder, known as, I know it's a very difficult thing to pronounce, I can't even do it right, Pelizaeus-Merzbacher disease, or PMD for short. Conatal PMD is the form that's being treated here. It's an invariably fatal childhood illness. And the problem is that the children just don't have functional oligodendrocytes. These are the cells that insulate nerve cells. And they're born with a metabolic defect that really gives them no functioning oligodendrocytes. So once again, it's an attractive candidate for cell replacement therapy. If you can introduce into the brain functioning oligodendrocytes, you know, that will actually do the work that the native ones are not doing, you might actually rescue these cells, actually rescue the children. So a first step in that direction is to take neural stem cells that have the ability to make oligodendrocytes. And in mice, it's been shown that these cells, when they're in the brains of mice that don't make oligos, there's a mouse model of this disease called a shiver or mouse, where there are no oligodendrocytes. If you can take these human neural stem cells and put them into the white matter, where the oligos normally live in these mice, they'll actually make oligodendrocytes and make the myelin that's missing in those mice, and you can actually rescue the mice. That is, these mice, instead of dying and shaking and so on, stop shaking and they live, you know, a normal lifetime with these human oligodendrocytes making myelin for those animals. So that proof of principle study was enough to persuade the FDA to authorize a trial using the same approach in patients. So this is a safety trial. It's a phase 1 trial, and the idea is to see, you know, what the problems will be, not so much what the benefits will be. And that trial has been approved by the FDA. It's a collaboration with Stem Cells, Inc., which is a company here in Palo Alto that makes these cells, and David Rowich, who's a pediatric intensivist here at UCSF, is heading that trial. And so, again, the strategy is to use these neural stem cells that are capable of making oligodendrocytes. This is a schematic of one of these oligodendrocytes. It has all these little processes. Each one of these wraps around this myelin, forms the myelin, and insulates these nerve cells. And the hope is that by putting these cells into the white matter of these children, they'll make myelin, they'll insulate some of the neighboring axons, and we'll be able to see that in MRI scans, because MRI scans are pretty sensitive to myelin. And in these children, there's no myelin, so it should be possible to see if the strategy is working in life. And again, this is only possible because there's a way of making oligodendrocytes from neural stem cells. We can do that in the dish, but it turns out that the body has a pretty good way of doing it on its own. And so the strategy here is to actually put the neural stem cells. Now these are, you know, not pluripotent. They can't make heart, muscle, and bone, and so on. But they will make oligodendrocytes. They'll also be able to make nerve cells. But the animal studies have shown that if you put these in the white matter, they don't make nerve cells. They make oligos most of the time.